Up next, we have Josh Sommer and Joan Levy. Going to shift a little bit from the spirituality to the research. Josh Sommer, our executive director, I don't know that I need so much introduction for him. I think you all know Josh. And Joan Levy is our director of research and has recently joined the foundation a few months ago and has accomplished so much in that time. And Josh and Joan work closely together, and so they're going to give their presentation somewhat together in tandem. So welcome, Josh and Joan.
Well, good morning, everyone. Good to see you all back here this morning. So just reflecting for a moment on Deborah's talk, it was occurring to me that I've often thought over the last 11 years now, it's hard to believe almost 12 years since I was diagnosed, that Chordoma is a club that really none of us choose to be a part of. I don't think any one of us would have chosen Chordoma, would have actively wanted to be here, but I think for me personally, it has turned into an opportunity to find so much meaning, to explore so many different parts of the world, the country, to be exposed and to meet so many amazing people like all of you in this room, to have a chance to work with a truly extraordinary community of doctors and researchers who are coming together with such great determination and a sense of collaboration and determination and spirit, and it's really been an incredible journey. And I think, frankly, many of you in this room have become my closest friends and colleagues, and that is really a special thing. And so I am really grateful to all of you for helping to make meaning of this situation, and want to especially thank the doctors who have enabled me to be here, my team of surgeons at the University of Pittsburgh, and Dr. Gardner is actually here from the University of Pittsburgh, and I truly can't thank you enough for giving me these last almost 12 years. One thing I'm struck by is that every year when we get back together, I feel this increasing sense of hope. I think that we look back on where we've come in the last several years, and every year I think we just kind of, our eyes kind of get wide, because the progress that's been made in the last year kind of makes the previous progress pale in comparison. And so it's always really exciting and energizing to get to speak with you all on the heels of the research conference, because there is just so much happening. We were joined yesterday by 140 researchers from 12 different countries, representing essentially every specialty involved in the treatment of chordoma, researchers from across the scientific continuum, from basic researchers to drug developers to clinical researchers. I think one thing that's really, really important to note is that for the first time we had 12 different drug companies that were present yesterday. That is an extraordinary, extraordinary turn of events. I think when we started, I think as many of you know, there were essentially no companies that were interested in chordoma, and so that we have gotten to the point now where the field has progressed to the point where 12 companies are involved is really, really an extraordinary feat. So I want to talk about what the foundation has done and is doing, and reflect on some of the progress that's been made, and then, as Shannon said, welcome up my colleague Joan Levy, our new director of research, to talk to you about where we're going in the future. Okay, so I want to start off with the premise that we talk about accelerating the search for a cure, but I actually want to talk. I want to take a different approach. I think when I was first diagnosed, and when the foundation first started, and I think when many of you first started coming to these conferences, we talked about needing to find a cure for chordoma. But I've increasingly come to take an optimistic view and realize that, in fact, chordoma is curable. It's just that it's curable in some people, and not all currently. It's curable in those people if they get the right care, if they get the right initial surgery or radiation. And we're learning that surgery and radiation have improved greatly over the last several years, last decade certainly, but in particular in the last several years. And then I guess the question is, at what cost? So sometimes surgery and radiation can lead to significant side effects, to significant impact on quality of life. And so recognizing these things, we as an organization and as a community, we recognize that our task is not just about developing better treatments, but helping all of us, everyone who's diagnosed with chordoma, get the best care possible so that we can all benefit from curative treatments that already exist. And our vision as an organization is for everyone, not just some people who have chordoma, but everyone that's affected by this disease to be able to overcome the disease and maintain our quality of life. That's really important. Not just to be able to live for a long period of time, but live for a very long period of time and have the quality of life that we desire. And I think that that is something that we as an organization and the research community has really come to recognize the importance of. So I think, as all of you probably know, when we started off, when the organization started off 11 years ago, how we realized this vision was very much an open question. We really didn't know where to start. We were starting very much from square one. There was so little known about this disease. There was virtually no one working on it. There were essentially no treatments being developed. So how do we go about realizing that vision of making chordoma a disease that we can all overcome with a good quality of life? So the first thing we needed to do is develop a plan. And to do that, as many of you know, we pulled together the chordoma research community, as it were, at that time, which was a handful of investigators from across the world. But we also thought it was really important to bring together experts, doctors and researchers who were studying other types of cancer and other diseases, who had relevant expertise, who could help us come up with a plan for how to solve this problem, how to attack chordoma. And it was extraordinary to see these dedicated researchers come together. It was just electric in the room to see all the ideas flying and to get such good feedback and such good ideas and a lot of questions, frankly, that needed to be answered. And what emerged from this first meeting back in 2007, back in Bethesda, was a clear sense of some of the key questions that needed to be answered and the key research priorities that would need to be addressed to move the field forward and ultimately find cures for everyone. What that turned into was a research roadmap that we as a community and we as an organization have been pursuing in the years since. And I want to just walk through it at a very, very high level to give you a sense of how this roadmap has progressed and is progressing. And it really starts off with, and I promise you I'm not going to get into a lot of nitty-gritty science here, but at a high level, I just want to share that it starts off with creating the basic resources that researchers actually need to be able to study the disease. This was a huge problem we faced back then. We didn't actually have the raw materials, the cell lines, the animal models, the tissue, the things that were needed to study the disease. Having those resources enables us to apply what are now really, truly extraordinary technologies to be able to understand the biology of the disease and to identify its vulnerabilities and its weaknesses, what about chordoma might make it sensitive to a particular type of radiation, to a particular type of drug? What can we learn about the biology of the disease to find an Achilles heel, to find some way to target or to precisely treat the disease while leaving normal healthy cells and the rest of the patient unscathed? Using that knowledge of what's driving the disease and its potential vulnerabilities, could we either find a drug that already exists or a therapy that already exists, or might we have to develop new therapies to address the specific vulnerabilities and the specific weaknesses of chordoma? We didn't know that at the time. The only way to know that was to understand the biology of the disease and let the biology guide us. Once therapies were either identified or developed, then there was a need to actually test them in models of the disease to give us a sense of whether they might work before taking them into the clinic and exposing patients to what could potentially be dangerous or toxic agents, figuring out whether we have strong reason to believe in laboratory models of the disease, whether in fact, whether and how these therapies might actually work. And then finally, where the rubber meets the road, is actually testing promising therapies in the clinic. Because at the end of the day, no matter how smart we think we are, the only way to know for sure whether a treatment is actually working or going to work is actually for patients to try it. There was a researcher that came to one of our early research conferences who was fond of saying that, look, you can study cancer in animals all day long, but at the end of the day, humans are the best models of human disease, and you can't lose sight of that. So the ability to test drugs in patients in clinical trials was absolutely critical. Now, where were we back then in 2007? There was one cell line. There were no animal models. There were no sources of tissue. So this made it really, really difficult to actually do meaningful research. And some of you in the room may know that before starting the foundation, I actually worked in a lab at Duke trying to study chordoma, and we faced these challenges firsthand. There were so many experiments that we wanted to do, so many ideas that we had, but we were really stuck because we didn't have access to these basic materials. So that was a real challenge that needed to be addressed, and needed to be addressed first, frankly, to be able to make progress in subsequent areas of this research roadmap. That's where we focused a lot of effort over the last number of years. Largely because those resources didn't exist, we knew virtually nothing about the biology of the disease. So we were truly starting virtually from a blank slate. We had some inklings of, as I mentioned last night, that maybe this newly characterized gene brachyury might play a role, but we didn't really know what that meant. There was no one, really no one, developing new treatments for this disease. There had been no drugs ever tested in animal models. And there had been one clinical trial that had ever been done. One clinical trial that had ever been done. So we were starting with a daunting task ahead of us. And the only thing that we knew to do was to take it on one step at a time. But we wanted to be smart about it. And so we wanted to try to figure out how we could be clever about doing this, how we could be efficient about doing this, and how we could optimize our chances of success. And so we did a lot of research as an organization. We looked to what other organizations had done. And what we realized was that there was, we faced a big challenge. We faced a big task. And we as a small, relatively small patient community, to raise the kinds of resources and to generate the kinds of investments that would be required is really an enormous undertaking. So, tens and hundreds of millions and billions of dollars are invested in cancer research every year. How can a small organization, how can a small community like ours have any hope of making an impact and making a dent in this disease? And we realized we needed to do things differently. So the traditional approach of many organizations like ours, many disease-specific organizations, is to raise money from members of the patient community, from events, from individuals, and to put out a request for proposals, and to try to get a lot of good ideas about projects that researchers might want to do, and then try to do your best to select those best proposals. And that's really important. But it's a passive approach, and it assumes that a field exists of researchers who are equipped to actually do all the meaningful research that needs to be done. But of course, in the case of Chordoma, we were starting off with such a small community, just a handful of researchers. That wasn't a safe assumption. We didn't have all of the expertise in the community that was needed to actually take us from the most basic research all the way to the clinic. So we recognized that we needed to be proactive and set a research agenda and go out and find researchers who are best equipped to take on the research priorities that the field identified and get them interested in Chordoma. We recognized that just funding grants was not enough, that we needed to create an ecosystem and an infrastructure to enable many more researchers to bring their expertise to bear on Chordoma, to create an environment that attracted researchers, attracted talent, attracted companies to this field, so that they would bring their own energy and capabilities and resources to bear on this problem. We recognize that sometimes there's more efficient ways to get research done than simply providing grants. While grants are very important, there were some problems that we found were actually more amenable to being solved by offering prizes to incentivize many different researchers to try their hand at solving a problem. And in particular, we applied this approach to developing cell lines, and I'll talk about it later, but that actually turned out to be enormously successful and has actually now turned out to be a model that other organizations have emulated. We've also employed the use of contract labs, where the foundation actually has contracted with research organizations to run our own experiments, and that enables us to generate data and make it available and share it in real time. And finally, we've recognized as a small community, we have to measure our success not solely by the amount that we raise and invest, but how much we can leverage from the NIH, from academic medical centers, from drug companies. So if we can invest $100 and we can bring in 1,000 from outside resources, that's how we're going to succeed. That's how we're going to get where we need to go as a small and scrappy organization and community. And I'm really happy to say that we didn't know if this was going to work. We didn't know how it was going to turn out, but I'm just so filled with hope and optimism to see this catching fire and to see us actually surpassing our hopes and expectations in many of the areas of this research roadmap. So, as I said, when we started off, we were suffering from a real lack of the critical resources that we needed to move the field forward. Now, over the last decade, we have essentially, we have come a long way towards solving this problem. So there are now abundant and well-characterized cell lines representing almost every subset of the disease. We are well on our way to developing a panel of well-characterized animal models to enable drug testing experiments. And thanks to many of you in this room, we've amassed a repository of biospecimens that are now available to the research community to fuel research. So it's impossible to overstate how much of a hurdle has been overcome in getting past these problems. This opens up whole new avenues, whole new fronts of research that were completely inaccessible to us when we started. As a result, the research field, as we've heard over the last several days, has just exploded. Many new researchers have been able to come into the field to apply their expertise to studying Chordoma, and this has led to a real explosion in new knowledge about the disease. That knowledge, some of it, sometimes when you learn things about the disease, not all of it is meaningful, not all of it is translatable, but about 20 potential therapeutic targets have been identified. These are things about Chordoma that may lend themselves to applying a particular drug to treat the disease. The great thing is a lot of those are what we call tractable targets. They are, or you could call them druggable targets. There exist treatments already that might be applicable. One of them in particular, brachyury, which we've talked about, is not yet druggable. And the biology has indicated that perhaps that is the most important or one of the most important targets. And so that leads us to our next task, and something Joan will talk about more. But over the last decade, this inkling that brachyury is important has really turned into a, I think, proof positive. There are so many arrows now that are pointing in brachyury's direction. So many investigators have independently come to this conclusion. So many layers of evidence suggesting that brachyury is the key driver, the master regulator of chordoma, and its chief vulnerability. And so we have, over the last couple of years now, begun, and I would say in a preliminary fashion, to sow the seeds to take on brachyury, to develop new therapies. And we have partnered with and funded a number of institutions across the country and across the world to sow those early seeds, to take on the foundational steps to develop drugs to target brachyury. And we just had a phenomenal session on Thursday, a breakout session that was led by our scientific advisory board member and now board member David Drury from UNC, to try to crystallize a very clear plan about how to move this forward in a systematic fashion. And I'm so delighted, as Andy mentioned, that we've forged this new partnership with the Mark Foundation to spur more research in this area. And we hope to invest very, very significantly, as Joel mentioned last night, over the coming years in really taking on this problem head on. And I should say in a concerted and coordinated and team effort involving the institutions that you see there on the screen as well as others, and importantly, involving companies as well. And there are already a number of companies that are becoming interested in brachyury. As we've taken on brachyury, in parallel, we have recognized a need and an opportunity to test drugs that already exist and drugs that are in the pipelines of various drug companies. And so without getting into all the details, I'll just say that this turned out to be a very, very logistically complicated endeavor. We tried to do this by offering grants. That turned out to be very, very costly. To do this in academic labs turned out to be very, very costly, very slow, very inefficient. Some of that has to do with just the way that academia works, and a lot of it has to do with the fact that chordoma is very, very slow growing, and so these experiments are long and costly and difficult. So we said it makes no sense for every lab to be replicating these same experiments. Let's build a shared infrastructure. Let's build the equivalent of an interstate highway system that we can all use, and that we can all use freely, and equally, give everyone equal access. So we've built a centralized drug screening program that's operated by a contract lab, such that any researcher with a bright idea that gets vetted by our scientific and medical advisory boards can test their idea in models of chordoma and very, very quickly find out whether there's a good chance that it's going to work, whether it seems to be effective in these animal models. And I just throw up one chart which represents data that's come out of this drug screening program. And this, by the way, is part of the data package that provided justification for the clinical trial that we heard about from Kevin Mellert, who came over from Germany last night. So the group at Ulm generated this hypothesis in cell lines and tissue. We were able to test their hypothesis in animal models of the disease and generate a very impressive result. So you can see the blue line is animals that are treated with palbociclib, this drug that was found by this group in Germany. So as you can see, it really significantly slows the growth of chordoma in these mice, and has given us a great deal of optimism about the chances for this drug to be effective in patients who have the particular molecular alteration that would render their tumor sensitive to palbociclib. So I want to not raise hope, undue hope or undue expectations. This may not work for everyone, and of course we know that mice are not people. So take this with a grain of salt, but it is very encouraging preclinical data that gives us a lot of hope and optimism for the clinical trial. And then finally, we have kind of gone from this place of not knowing whether a clinical trial could even be done feasibly in Chordoma to working with our medical advisory board, and I want to in particular recognize Dr. Shreyas Patel, the chair of our medical advisory board, as well as many members of our research community for helping us essentially understand the challenges that were standing in the way of clinical research and devise a set of programs to help overcome those challenges, to do what many people have traditionally assumed has been impossible. So when we first started talking about doing clinical trials for Chordoma, most people we talked to said, how on earth are you going to run clinical trials in such a rare disease? This is what keeps companies out of the rare disease space. This is why companies historically have neglected diseases like Chordoma, because there are such significant challenges to running clinical trials. But we have developed a series of programs to be able to support clinical trials in a number of ways, financially, with the expert input of our medical advisory board to help design trials to optimize their chances of success, and importantly, to be able to connect patients with trials, and that's a big part of what Shannon and her team do. And I'm really, really happy to share that now we have gone from having just one clinical trial to a pipeline of 7 clinical trials that are either started or getting underway. And this is really, for me personally, a true source of hope. And I think the perhaps even more hopeful thing is that what we heard over the last couple days is that this is just the tip of the iceberg. There are several more trials that are kind of on the heels of these. Including this afternoon, our Medical and Scientific Advisory Board are actually discussing and vetting proposals from 6 additional groups who have submitted trial concepts. So 6 more groups interested in running clinical trials in Chordoma. That's compared to 1 when we started 10 years ago. So I think that's, more than anything else, I think that just conveys the sense of scale as to how things have changed in this disease. And because we can now run clinical trials, because we can now efficiently do research across that entire therapeutic development continuum, Chordoma is no longer untouchable from the standpoint of a drug company. It's no longer this disease that is kind of hopeless and impossible to work on. In fact, now Chordoma represents a big opportunity for companies because it presents a combination of 1, a significant unmet need, 2, a very motivated and unified research and physician community, and 3, a very active, engaged, committed patient community that is motivated and connected and able to participate in clinical trials. And that represents a big opportunity for these companies. So having gotten to this point, I'd say having cleared away many of the obstacles that stood in the way of our progress initially and made Chordoma a viable place for companies and top researchers to do their work, there now is a lot more research to be done. There's a lot more that can be done and a lot more that needs to be done than when we first started. The possibilities, the universe of possibilities has really expanded greatly. And to take advantage of those possibilities requires more on the part of the foundation. It requires scientific leadership, scientific expertise, a lot of coordination, a lot of hard work. And so one of the things that I am most encouraged and most happy about over the last year really, is that we have been able to recruit for the first time a extraordinarily accomplished, seasoned and expert cancer scientist, Dr. Joan Levy, to be our first director of research. And I just want to quickly give a snapshot of Joan's background, which is so incredibly impressive. Joan is a unicorn. When we set out to find someone to fill this role, we wanted someone who had experience across academic research, across industry, and across across philanthropy and a patient-driven organization. If you draw a Venn diagram of the people in the world who have that experience, there's probably 1 or 2 or 3 people in the entire world who have that experience. Joan Levy, to our great fortune, being one of them. Joan has had a very illustrious 30-year career across each of those critical areas of research. First, as a cancer scientist at Yale. Next, as a director of a research unit at Bayer Pharmaceuticals, where she developed cancer drugs, including, ironically, some drugs that may be relevant for chordoma. And then, for the last 10 years, serving as vice president of research at one of our peer organizations, and arguably one of the premier patient-driven organizations in the entire world, the Multiple Myeloma Research Organization. And over the course of Joan's tenure there at the MMRF, there were 10 drugs approved for myeloma. And Joan can tell you, I think there was 1 or 2 approved when she started there. And Joan will be the first to tell you that this was a big team effort, and she's not going to take credit for all of it. But she had a significant role to play. And I said, Joan, when you get 10 drugs approved for chordoma, then maybe you can retire. Exactly. So it's my great pleasure to introduce our new director of research, Dr. Joan Levy.
Wow, what a great few days, and I'm so happy to be here. And I have to say, just to reconfirm what Josh said, I have worn many different hats in oncology research, but the one that's most special to me was the time that I spent at Oncology Foundation, the 10 years at MMRF, because it enabled me to interact with the people that matter the most, the patients and caregivers like yourself. It's a foundation that enabled me to do that. And therefore, it's very special that I was able to be given this great opportunity last June to join really the leading rare cancer foundation, and to join the exceptional team at the Chordoma Foundation, as well as the incredible clinicians and researchers that do chordoma research in order to help find solutions for chordoma. So I'm thrilled to be here. I'm so happy I was able to interact with many of you last night, and I will continue to interact with you more as the day progresses. So what am I here to do right now? I'm here to help to talk to you about our priority research areas. You've heard about many of these from Josh, the first being to develop these first therapies targeting brachyury, to apply immune therapy advances to chordoma, and most importantly, and most relevant to all of you, to accelerate clinical evaluation of promising therapies. So what I'm going to do now is just go very briefly into each of these 3 areas, and I'll start off with brachyury. So you've heard that word, and maybe I'll have to turn on for a minute my kind of nerdy science hat to explain to you what brachyury does. There's a picture of it, and what you see there in red is brachyury. It's a protein that sits on DNA, the coding material of life, and regulates the expression of different genes. It's a member of a protein family called a transcription factor, and brachyury as a transcription factor regulates the expression of genes in chordoma cells, and it drives the growth of those chordoma cells and causes the chordoma cells to survive. So therefore, not surprisingly, you want to turn brachyury off, so you will downregulate that growth of chordoma cells. And interestingly, brachyury is not only important in chordoma, it's also important in the progression and metastasis of other cancers, such as lung cancer, breast cancer, prostate, as some examples. So, as Josh mentioned, we have supported research, and there's an international team. We supported research on showing that brachyury is the key driver in chordoma. And this has attracted that great international team that's working on trying to find emerging therapies for brachyury. However, brachyury is what we call an undruggable target. It really has a conformation that's really hard to find drugs. But I'm going to be optimistic here, because there are so many emerging technologies, especially that we've heard about lately and at the brachyury session, that really will help develop therapies against targeting this very difficult target. And we actually supported 2 of these research programs last year through generous donations, 2 grants to Dr. Slim Sassy and Dr. Charles Lin that are applying some of these emerging technologies to the study of brachyury. And finally, as Josh mentioned, we do have a new and wonderful partnership with the Mark Foundation for Cancer Research to fund innovative therapeutic strategies. It's going to be a win-win, because targeting brachyury will not only be important for chordoma, it'll be important for other cancers that are driven by brachyury. And finally, what I haven't mentioned is that brachyury as a transcription factor, there are other transcription factors that drive cancers. So if we're going to be able to figure out how to target and drug brachyury, the oncology field will be able to figure out how to target other transcription factors that are key drivers in other cancers. So this is a beautiful and wonderful opportunity for us. Okay, next research priority, advancing immune therapy. I mean, immune therapy has made a renaissance in oncology. So many new treatment, immune therapeutic approaches that have been approved in a number of different cancers. I mean, how many of you read about immune therapy almost weekly in the New York Times, hear commercials about different immune therapies? I mean, it's constantly being discussed. And we want to apply those immunotherapeutic advances in other cancers to chordoma. But first, we have to answer some very basic questions. How do immune cells interact with chordoma cells? And you might think, well, chordoma cells, aren't they kind of foreign? Wouldn't the immune system attack them just like they attack bacteria and viruses? Well, presumably tumor cells like chordoma cells have figured out a way to escape the immune system. In addition, we need to figure out through our target discovery programs, are there specific immune targets to chordoma that we might be able to drug? And leading this into the clinic, how do we design the most beneficial immune clinical trials? It's probably not going to take one immune therapy to be able to combat chordoma. It's probably going to take combination approaches to be able to combat chordoma as well as other cancers. And finally, the big question that everyone might think about is, why do some patients respond to immune therapy and others do not? That's a really important question. We have to take those samples of patients who are on immune therapy drugs and in trials, and we have to figure out why the immune cells in those patients are working or not working. And that's a huge question in immune therapy research as a whole. We had a wonderful immune breakout session the other day that addressed many of these questions, and now, in the next few months, we're going to assemble the leading immune therapy key opinion leaders to be able to offer a request for proposals to the research community to identify meritorious applications to advance immune therapy in chordoma. And I'm very excited to do that. This was a field that I have always had passion for, also in my previous life at the MMRF, and I certainly will have the same passion for advancing this in chordoma. Finally, our last very important research priority, near and dear to everyone's heart, Josh mentioned it before, clinical trials. And we are building our clinical trial pipeline There are 7 trials that are chordoma specific that we support in many different ways, as Josh mentioned, either funding, through patient services, increasing awareness, what Shannon's group does, as well as working with our medical advisory board for their expertise on designing the most important and the best possible protocols for chordoma patients. And we do have 3 trials that are currently enrolling patients. 2 of them, the first 2, are, one is a vaccine trial, the GI 6301, and then we have a trial that's testing an immune checkpoint inhibitor. That's the one, nivolumab. The Opdivo is the same as Opdivo that you hear on commercials. That has been approved in other cancers. We are applying that and testing that in chordoma. And finally, as Josh mentioned, we have a palbociclib trial. That is not an immune trial. It's a trial against a targeted protein. And again, we're so pleased that both that preclinical research at Ulm in combination with clinical expertise at Heidelberg was able to be combined to bring basic research findings to the clinic and enable this trial. And this trial is currently open and enrolling patients in Germany. And it's not surprising, many of you were at the dinner last night, that the 2 groups won the Uncommon Collaborator Award. And finally, we have a trial very near to opening. It's called afatinib. It targets a special protein called epidermal growth factor receptor that's overexpressed in a large proportion of chordoma tumors. And that trial is soon to open. I will say that some of the research for that trial was generated by us in our drug screening program, and it was recently published a few months ago. And we are expanding the clinical trial pipeline. What is my job in this? My job is really to focus on this year speed in opening these trials and speed to accrue patients, working with Shannon to really increase patient awareness, being on top of these trials and opening others. And I would like to say 2 other points, actually 3 other points. The first is, in terms of the clinical trial pipeline, we have to view it like an investment portfolio. We might not want to put all of our eggs in one basket into one stock, so we need to be able to open trials using different agents, these targeted agents against specific proteins, different types of immune trials. Not every immune agent is created equal. There are certainly very different types of immune therapeutics out there. Number 2, we need to open these trials worldwide, across the US and internationally, so chordoma patients worldwide can have access to these agents. And finally, what I want to state is that what we need, that I don't want to leave you with the impression that these are the only 3 trials open for chordoma patients. These are chordoma-specific trials, but there are a number of trials that are open in solid tumors that chordoma patients can join. Information on those trials is in our clinical trials catalog that can be found on our website, as well as speaking to Shannon and patient services, as well as myself. So again, we're committed to these 3 programs. I'm not going to go over them again, but I'm going to conclude by saying, as Josh said, we are working with an incredible number of researchers. I mean, I can't begin to tell you what this conference was for me. It was my first one. And the passion in that room from the clinicians and researchers, the amount of material that was covered was just incredible. It was an extraordinary workshop, very productive. There were a number of companies present, and that was incredible. But in addition to all that, we are developing new opportunities with leading organizations. I mentioned the Mark Foundation for Cancer Research. We also, through a new funding opportunity that we're very grateful for, have an opportunity to offer grant programs in Canada through the Canadian Cancer Society. And we're working with a number of other organizations, including some from the government. I just want to mention one. in particular, we were privileged the other day to have a pediatric chordoma breakout session. And at that session, Dr. Brigitte Wideman was the moderator of that session. She is one of the world's leading experts in pediatric oncology. And all I can like to close is she came up to us afterwards and said, and she mainly does other types of pediatric oncology, so chordoma is new to her. She came up to us and said, this is unbelievable. I want to go back and think about what I've heard here and how I could apply it to chordoma. And hearing this from people, attracting new people into the field, was music to my ears, especially of her caliber as well as many others. So I want to leave you with, I'm thrilled to be here. I will always be available for all of you. Please enjoy your day, and I hope to interact with you over the course of the day.
So I think you get a small taste of why we are so excited to have Joan on our team. This is truly a quantum leap for us at the organization and is just unlocking so many possibilities. So Joan, thank you. That was really phenomenal.